Welcome, MSM, to our second MSM Journal Club meeting. My name is Yazan Bouchi.
My name is Vaghani.
My name is John Woody.
And I'm Crystal Uwazie.
And today, we're presenting a very odd case of a stem cell transplant.
Go to the next one.
But we'll start with the background.
Okay, so what is HIV? Anybody know? HIV, human immunodeficiency virus. There are two main subtypes, HIV-1 and HIV-2. Morphologically, they are pretty much indistinguishable. However, they come from two different types of animals. One is a primate, one's a monkey. As most of us know, it's transmitted primarily through bodily secretions such as blood, breast milk, semen, and vaginal fluids, and is transmitted from host to host through mucous membranes. In the United States in particular, it's known to disproportionately affect African Americans and members of the LGBTQ community. And while at one point it was a very serious and still is a very serious disease, it's no longer considered to be a death sentence because there's so many forms of treatment, such as antiretroviral therapy, pre-exposure anaphylaxis, and post-exposure anaphylaxis. So how does HIV work? How does it create immunodeficiency? So the virus, usually what it does is when it enters into the body, it attaches to CD4 molecules on helper T cells, as well as CCR5, which is a GPCR, which is normally present in immune cells. The function of CCR5 is not really known. However, it is known that whenever HIV binds to CD4 and the CCR5, it helps it to fuse with the membrane and enter the T cells. After it enters the T cells, it uses reverse transcriptase, which is characteristic of most retroviruses, to integrate itself into the host genome, and then uses the host cell's own organelles to basically produce its own structural proteins. Once these proteins are produced, it helps it basically put itself together into these viral buds, which then are released into millions of HIV particles, which can then later on go to infect other CD4 T cells. The end result in these patients is typically a massive reduction in CD4 T cells, which is what AIDS is, acquired immunodeficiency syndrome. When the CD4 count becomes lower than 200 cells per microliter, that leads to very severe autoimmune issues. So who is the London patient, the subject of this paper that we're presenting on? So this is participant number 36 in the ICI STEM cohort. This cohort is specifically, as the name suggests, it's an organization that uses stem cells to try to cure HIV through stem cell transplantation. This patient had hematopoietic stem cell transplantation using that had a deletion mutation in CCR5. And these cells were transplanted for treatment of Hodgkin's lymphoma. After this occurred, they saw remission of the HIV as evidenced by undetectable levels of HIV in the plasma and in the peripheral T cells. So basically, the subject of this whole paper is whether or not this constitutes a cure for HIV. And just kind of as a side, there's only one person ever that's been cured in this manner, which is an individual known as the Berlin patient who underwent a very similar stem cell transplant for the treatment of acute myelogenous leukemia. Now, methods and findings.
So this is just going to go over the different tests that they did to determine whether or not they cured this patient of HIV-1. So the first set of testing was for viral load. Essentially, this is just testing how many virus particles are in a specific volume of bodily fluid. This was done with two different assays. One was a hospital-based HIV-1 viral load assay, and another was an in-house ultrasensitive plasma and cerebral spinal fluid viral load assay. The reason why they did both a hospital-based assay and an ultrasensitive CSF assay is because viral replication can occur in CSF even if a patient is on retroviral therapy. So they just wanted to essentially cover their bases. And what they found out was that there was essentially no replication of the HIV-1 virus. There was less than 1 copy per ml. Next, they also did blood and tissue process. What they did with the peripheral blood, what they did with peripheral blood was isolate PBMCs, which are essentially your blood minus red blood cells. So essentially all of your white blood cells, immune cells. And this was done via a Bicol density centrifugation procedure. And they also got biopsies from different tissues. This would be gut tissue and lymphoid tissue. So what they did was got biopsies, then extracted DNA from those biopsies. So they used 2 different types of PCRs in order to actually study the DNA from the different tissues that they were studying. So they did both RT-PCR as well as what is it?
qPCR maybe?
Droplet digital PCR, abbreviated as RT-PCR for quantitative real-time, and then DD for droplet digital. From what I understand, droplet digital is a more, it's just a more, I guess, specific test. It gives you better, essentially, results in the sense that it's more of a quantifiable sort of form of PCR. And RT-PCR, I feel like is more, you see that more in studies. It's kind of like the typical PCR that people run. It just uses fluorescence technology, whereas the ddPCR uses some sort of water, oil, emulsion technology. So these are the findings from the RT-PCRs that they ran. Thank you. So what they found in their gut biopsies was no detection of HIV-1 LTR or long tandem repeats. In the lymph nodes, there was no detection of HIV-1 LTR. And in the lymph nodes, they did find HIV-1 envelope and psi being expressed in the cells. For the ddPCR, what they found were in the gut biopsies, no detection of HIV-1 LTR. And in the lymph nodes, no detection of HIV-1 psi. And they did detect HIV-1 LTR and envelope in the lymph nodes. So they also studied T cell responses. This was done via flow cytometry and cytokine staining intracellularly. They looked at CD8 and CD4 T cells. And essentially, this was just to see whether or not HIV was replicating in the body at all. And the most important thing to really look at is, there's an animation. Yeah. So if you compare the HIV GAG stimulated cells, essentially what they did was stimulate PBMCs, their T cells with a protein that's involved in HIV-1 proliferation. And they wanted to see whether or not there would be some sort of immunologic activation of the T cells when they did that, which would indicate that there was proliferation of HIV-1 in the body, if you did see that result. But you can compare the HIV-1 GAG stimulated cells to the unstimulated cells, and you can see that similar percentages, which are what those numbers are, of cells were actually producing interferon gamma compared to the control, essentially. So there was no marked increase in the production of interferon gamma. And then if you look at the CMV-PB65, this was, if I remember correctly, this is cytomegalovirus, a structural protein. and they use that as a stimulant for their cells. And as you can see, when they stimulated cells with that, there was a marked increase in the production of interferon gamma intracellularly. And that's just another way you can kind of compare the results between what an increase in interferon gamma production would look like. So next would be antibody responses that they looked at. This was just to study the antibodies that were being produced by the London patient. They used ELISA and Western blot. This is just two different methods that you can use to study proteins in a sample. And what they found were when they did these different tests. So with the standard ELISA, they saw that 2 years after the retroviral therapy, 2 years after the transplant of the hematopoietic cells, there was production of antibodies that were against HIV-1. But when they did low sensitive ELISA, as well as the Western blot, they saw that the antibodies were decreasing over time within that 2-year span. So that's what these results are essentially showing.
Okay, so we'll move on to the diagnosis. So any guesses of what the diagnosis of this patient is? Any takers in the crowd? Anyone on the panel?
Thumbs up HIV, thumbs down HIV.
Thumbs up, he has HIV still? Okay, so let's go.
Still? I don't know.
That's a big question. So HIV or not? So what they did find, so these are going to be more of a summary of their findings. And it's going to be, yes, basically, did they have a recovery or not? And so for CD4 T cell recovery, they noticed a markedly increase. So basically the idea was they did that stem cell transplant, stopped any treatment. And then, so this 30 months after, they noticed a markedly increase in those CD4 T cells. Then they did a viral load monitoring, like Crystal mentioned. They noticed decrease in those HIV in those fluids, such as the CSF, blood, semen fluid. They did a tissue and fluid analysis of both of them, yeah. And they noticed a decrease in those HIV molecules in that. And then finally, in HIV-1 DNA presence. So this was, first off, they said they took a biopsy and they said they found still some of those envelope proteins and those other things that were left behind. But they mentioned that these didn't have a big enough impact. And next. Oh, one more. There we go. So actually, yeah, so they noticed that it wasn't much of a concern and the levels were low enough that it would be considered undetectable. And so basically, our London patient has been in remission at the point of this paper for 30 months, which is amazing. Now, one thing they did notice, and I don't know if you, but the big question here is that chimerism of the stem cells. And up to this point, it is they calculated based on their models that he has a 38% chance of having a full remission. Now, if that chimerism of those stem cells integrating into the body increase, then that remission percentage can go up. But they will have to see based on that.
Yep. That was mathematically modeled. So what are the takeaways? Well, to start, the London patient is a patient who underwent a stem cell transplantation. And, stem cell known for not expressing CCR5, that's an HIV receptor. They did viral load assays for HIV-1. They did it for 30 months post-treatment, and they yielded no evidence of active viral replication. Sustained absence of this replication competence suggests a cure for HIV-1. That's pretty surprising. And this case demonstrates that a long-term, remission from HIV-1 is achievable through the CCR5 delta 32 allogenic hematopoietic stem cell transplantation is possible. This presentation was brought to you by Yog, John, Faiza, myself, and Crystal. Thank you guys for tuning in. We're going to take a quick intermission and have our next presentation coming up soon.